All right, so in the last video, we talked about water movement, right? We said that water moves by osmosis, right? Which is simply a fancy way of saying it diffuses through the membrane, right? So as the phospholipids shift, water can move through. Water's not the only molecule that can do that, right? You can get gases like oxygen, carbon dioxide. Some small steroid hormones can also do that. They can get through those shifting phospholipids. So if it's moving through phospholipids, it's called diffusion, unless it's water, and then it's called osmosis, right? Okay. Now, I mentioned in the last video that anything big or charged is going to need the help of a protein, right? And the word facilitate that you see here, facilitate, is a fancy word for help. So now what we're going to talk about is not the small stuff. Now we're going to use a protein to help things that are larger or are large and have big charges on them to get through the membrane, okay? Now, again, this is still a form of passive transport. So we're still moving these molecules with a concentration gradient, the same way we did with water, the same way we did with oxygen, carbon dioxide, small hormones, right? But now we're moving things that have a charge, and those things, or they're just too big and they can't get through the membrane. So here what we're doing is letting proteins help us to move things passively across the membrane. So that means we're still moving with the concentration gradient. We're still not using energy. Okay, those two things really important. All right, so some proteins can actually change shape when molecules that need to transport bind to them. Okay, so sometimes an ion can actually bind temporarily to a protein, and that will cause a conformational change in the protein that allows the thing to get inside. Okay, that's what we call having a carrier protein. So that's going to move charged and large polar molecules through the membrane. Okay, so that's a carrier protein that can do that. And then you have proteins that are called channel proteins that are like little tunnels that connect the outside of the cell with the inside of the cell. And those are things I think I told you, some of them might be aquaporins that we thought were just plain channel proteins, but actually can move water around the cell. But channel proteins just pretty much let stuff go through. Now, that doesn't mean there's no binding. It's just that any binding to the channel doesn't cause a conformational change. So this is always with a concentration gradient. Okay, so glucose, for example, can get into a cell through proteins in this way. All right, so here's the protein channel at the top, and notice this is cut away. So if the protein had a hole through the middle of it, you're seeing it cut away, but that's pretty much it. There's the channel protein. Here's the solute that we're trying to get through, like a big glucose molecule or something, and it's just moving through the channel from where it's highly concentrated to where it's less concentrated. Okay, and again, that's why it's passive. You're moving with the concentration gradient. No energy is required. Same thing here, except now our carrier protein actually binds to the molecule or ion, whatever it is we're moving in. Again, you still have a higher concentration of this solute out here than down here. So once the binding occurs, the protein changes shape, allowing the solute to get in. Super simple, right? Simple diffusion, except you're using a protein to help you to do it. That's why it's called protein facilitated. Now, sometimes we have to move molecules against concentration gradients. And for that, we use something called active transport. And active transport is going to use energy because we're either pumping it against a concentration gradient or we're having to take extraordinary measures to get the thing into the cell. Okay, so those are the two times that you would use active transport: moving against the concentration gradient or having to actually pinch something off into a vesicle and bring it in. That's what I mean when I say extraordinary measures to get the thing into the cell. All right, so the classic case of active transport would be moving things against a concentration gradient. And what that means now is that you're moving solutes from where they're less concentrated into where they're more concentrated. In other words, you have to actively pump the ions, and that's going to require energy. So now our protein is going from being a carrier protein to a protein pump. We've given it an active name. So oftentimes it's going to use a protein to do this. So the sodium potassium pump is a great example of one of these active transport pumps. It's an ion exchange pump, and it's going to change sodium for potassium so that you're moving ions to where they're more concentrated. However, also don't forget that endocytosis and exocytosis are also active transport. Okay, so endo, that root word means into or inside. You take macromolecules and other particles in by actually the membrane actually reaches out and pinches them off into a vesicle to bring them in. So when a cell takes in cholesterol from the blood to use it to create a membrane, that's one example. There's also exocytosis. So when the Golgi apparatus creates a package of proteins in a little vesicle, that vesicle can fuse with the membrane of the cell and open up and dump. You can also release wastes in that same way. It doesn't have to be from the Golgi. It can be a food vacuole that has had digested food in it that now fuses and dumps the waste out. Okay, so the pancreas releasing insulin, that's an example of an active transport via exocytosis. All right, so here is what the difference between these two types of transport looks like. Okay, notice these three are passive. So there's diffusion. If these little purple molecules were water, we would call that osmosis. But again, here, no proteins involved. You're just moving past shifting phospholipids. As they shift, water, gas just sneaks through. Passive transport using an ion channel like you see here, or a carrier protein, just involves passing through a protein to get in. But again, in all of these cases, notice you have the most concentration of whatever's being moved on the outside, the least concentration inside. So you're moving from high to low, which is with the concentration gradient, no energy. And then we reach this diagram. This is a diagram of the sodium potassium pump. So here you have these little ions, these diamond-shaped ones inside the cell. There are fewer of them outside, and we're trying to move them out. And so when they bind into the cell, or into fewer of them out here, more of them here. When these bind in, you get a conformational change that causes them to be pumped inside. Then once they're inside, these little orange ions can bind into the protein, causing a conformational change to move them out. And that's active because you're moving both ions against the concentration gradient. All right, and then here is what endocytosis and looks like. Actually, these are not exocytosis, but these are all endocytosis. Endocytosis can actually be split into three categories. Phagocytosis is where you are bringing in a solid particle. Okay, so if you have, say, a white blood cell that's bringing in a little bacterium, that it's going to use this method. So here's the bacterium, here's the membrane of the white blood cell reaching around and grabbing hold of the bacterium and pinching it off into a vacuole or a little vesicle. Okay, now this can be fused with a lysosome that would digest it. And then once this digested particle had its nutrients absorbed, then this little vacuole now is full of waste, and this vacuole can make its way to the membrane where it can reverse this process, and that would be exocytosis. Again, pinocytosis, you're bringing in liquids now, and they're dissolved solutes. So some liquid, some water with some solutes in it gets pinched off into a vesicle here. Okay, so literally phagocytosis means cell eating, pinocytosis means cell drinking. And then you have this receptor-mediated endocytosis, where receptors on the surface of the membrane receive molecules, and they all sort of migrate with the shifting phospholipids to a general area where these proteins can actually cause the membrane to pinch off a coated vesicle. they're all, as far as College Board is concerned, they just want you to know endocytosis and exocytosis. Whoops. Okay. So, and again, all of these can be reversed so that you can take a vesicle, fuse it with the membrane, and let it dump its contents to the outside, and that would be exocytosis. So these are endocytosis, reverse it, and you get exocytosis.